My name is Carmen Ruiz. I am from Peru. Today I am going to talk about isolation, identification, cloning, and phylogenetic analysis of microalgae Prorocentrum minimum. Pavillar Schiller, 1933, isolated from the Bay of Callao, Peru. This presentation is divided into 1. Objectives, 2. Introduction, 3. Methodology, 4. Statistical analysis, 5. Results, 6. Discussion, and 7. Conclusion. 1. Objective. Identify the microbiology Procentrum minimum from a water sample taken in the Bay of Callao, Lima, Peru. 2. Introduction. Some potentially harmful microalgae have caused fish mortality and fish egg, which have been associated with effects caused by incomplete decomposition of organic matter, anoxia, and external sources of marine pollution in phytoplankton species, mainly in natural fan shell banks. However, it cannot be ruled out that these species may become toxic due to environmental change of eutrophic processes. Eutrophication is the enrichment of seawater by nutrients at a faster rate than removal, causing substantial change in the balance of ecological ecosystem. The water is enriched with oxygen, causing the increment of phytoplankton and accelerating addition of nutrients. For this reason, high concentration of 1 more million planktonic microalgae cells per liter are observed. The color of the seawater will depend on the microalgae. In many cases, it can be greenish, brown, reddish, orange, etc. Harmful algal blooms is the term adopted by the Intergovernmental Oceanographic Commission of UNESCO and accepted internationality. 3. Methodology 3.1 Place of sampling. The study area was the Bay of Callao, exactly in front of the dock of the Instituto del Mar del Peru in Marpe. At 770936 W and 120336 S. It includes the constitutional province of Callao, as we see in Figure 1. 3.2 Isolation of the microalgae. We worked inside the laminar floor chamber in aseptic condition. A drop of the sample was deposited on a clean object slide and covered with a cover slip, being observed under the binocular microscopy in the 10x and 40x phases. It was verified the morphology and swimming movement of the leaf microalgae. The species of interest was observed. 3.3 Unialgae Purification Establishment. 3.3.1 Isolation by pipetting and successive washing with a sterile laminar flow to obtain a unialgae culture of the Procentrum minimum. A multi-well with 12 holes was used. 1 mm of molecular F2 medium was added with the sterile Pasteur pipette, and draw was taken from the first well and transferred to the second well. In this way, the successive washes were carried out. 3.32 Ground curve of the microalgae Procentrum minimum. The quantification of the microalgae growth density was estimated by the counting method in the 0.1 mm deep Neubauer chamber and with the help of the binocular compound microscopy at the 10x the report cell density. This is an effective method and correctly evaluate the daily cultivation process. Procentrum minimum cell count were performed daily for a period of 30 days. The assay was carried out the Phylon 1 control at the 19 degrees C and at the 21 degrees C. The objective of this trial was to observe how many days it took to concentrate the highest microalgae density. The control flags were not sampled daily for cell count, only the flags were sampled in the triplicate. 3.3% minimum concentrate for molecular analysis. Microalgae density was concentrated for 18 days at 21 degrees C. Then it was centrifugated at 5,000 revolutions for 10 minutes. The supernatant was removed and the pellet was refrigerated at 3 degrees C for molecular analysis. 3.3.4 Evaluate of the toxigenic capacity of the Prorocentrum minimum. 1.5 liter of the Prorocentrum minimum culture was used and it was centrifugated at 5,000 revolutions for 10 minutes. To the pellet, 50 ml of the acetone isolate phase was added, homogenized for 3 minutes, then placed on filter paper. Repeat 2 times. The acetone extract was steamed on the rotary evaporator with the water bath at the 40 degrees C. 3.3.5 Mouse's bioassay. I was inoculated 1 ml by intraperitoneal route of the liposolute solution of the extract of the microalgae Prorocentrum minimum figure 7 to 3 mice of 19 of 20 grams of the weight and it was observed for 24 hours. The bioassay was considered positive for the presentation of the toxin OA, DTX, PTX, and GTX when at least 2 of the 3 mice died within the 42 hours. 4. Statistic analysis. The normally and homogeneity of variance for the sample average analysis of cell density, cell milliliter for tenth. The analysis of variance ANDEBA and third-case test of cultivation temperatures and sampling days as factors. A level of signification was considered 0.05 for all tests and were used in Minitab statistical program 16 and system 13. 5. Result 5.1 point. Isolate of the microalgae. 2 strains of the microalgae were isolated Prorocentrum minimum isolated from Chucuito Beach in Callao Bay and Prorocentrum gracili isolated from Carpallo Beach also in Callao Bay. The isolation technique facilitated the work. Signs per culture were obtained from the algae unit. It is visualized in figure 7 and 8. 5.2 point. Ground curve of the microalgae Procentrum minimum and different temperature of 19 and 21 degrees C in the culture chambers. Is called be observed in the culture at 19 degrees C was nocturnal from day 25 to day 30, while the culture at 21 degrees C was persevered in the cell count from day 25 to day 30. Temperature is not a limiting factor for the develop and grow the Procentrum minimum, since this temperature fluctuates between 12 22 degrees C and salinities of 5 5.3 point Procentrum minimum molecular analysis. The result of the PCR was obtained by the combination of primer D1R and D2C and pairs ITS-S and the pairs ITS-8 for the Procentrum growth. The LSU sequence obtained corresponds to the species Procentrum minimum 2811 from the Probasoli-Gillard National Center of Culture of Marine Phytoplankton Institute and Procentrum minimum PM5, PM5.1 and PM7. Procentrum gracili PGR isolated from the Bay of Callao, Peru. 5.4 point statistical analysis. The highest cell densities of the microalgae Procentrum minimum were registered in the culture at 19 degrees C, shown in the figure 9. Cell density gradually increased with a significant difference until day 24 and 26 and then decreased. Culture at the 21 degrees C reached their maximum cell density at day 18, from which density decreased with significant difference during days 17 and 19. 5.5 point sequence alignment and phylogenetic tree construction. The sequence in figure 11 and 12 obtained from microalgae 2811, PM5 and PGR were aligned and phylogenetic trees were constructed to establish their identification and phylogenetic location confirm the species Procentrum minimum. 5.6 point result of the analysis of the mouse bioassay. The symptoms of the 3 mice after the inoculum of the microalgae were head down, with discomfort, blistering, hair and prostration. After 72 hours of observation, there was no death of the rodents. 7. Discussion. The result of the PCR was obtained by the combination of primer D1R and D2C and pairs ITS-S and pairs ITS-8 for PM5 and PGR crows. It was confirmed that these species belong of the Prorocentrum families and that maintain genealogical conserved regions. The three based on the LSU RNE analysis for Procentrum minimum proposed that the species are related to each other, where it includes PM5 and PM2811, considered as the species complete due to their similarity. It was concentrated the biggest quantity of microalgae from this. It was worked the protocol and indirect determination of the possibility effect of that toxin. It was negative, and one of the reasons could be the very long microalgae concentration of 10 for cells millimeters and 1.5 liters of volume filtrate to that density was not enough. 8. Conclusion. One of the main drawbacks of the sustainability of aquaculture is the presence of the reptiles, which can produce significant impacts on marine resources or cause economic losses. To face this problem, this investigation will contribute to provide the necessary information for molecular phylogenetic of the taxonomic diversity of the Prorocentrum minimum species of the Procentrale family present in the Peruvian Sea, which could lead to a possible event of algae blooms. Thank you.